Hello, everyone, and welcome to today's webinar, Complete and Comprehensive Biomarker Discovery, terraFlow for Flow Cytometry Analysis. I am Kaylee Bach of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by terraFlow Bio. To learn more, visit terraflow.app. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speakers, Pratip K. Chattopadhyay, PhD, Associate Professor of Pathology and Founding Director of NYU Langon's Health Precision Immunology Laboratory, Chief Scientific Officer of terraFlow. Also joining us today is Dan Freeman, Chief Executive Officer of terraFlow. For a complete biography on our speakers, please click on the Presenter tab. Pratip, Dan, thanks so much for joining us. You may now begin your presentation.
Well, thank you for that introduction. My name, again, is Pratip Chattopadhyay, and Dan Freeman's on the screen with me. We are the creators of terraFlow, and the tagline for today's presentation is never wonder what you're missing in your data again. We have been frustrated for years and years with how we do high-parameter, multi-parameter analyses, and we have come up with what we think is a really great solution that is complete and comprehensive. And Dan and I are going to tell you about that today. So we'll get started with the presentation. Flow cytometry technology has really exploded over the last 20 years. We've been able to easily achieve 20 and 30 parameter flow cytometry panels, and for that matter, mass cytometry and other types of cytometry panels. And the instrumentation, the reagents, the hardware is just beautiful. It provides intricate and beautiful detail about cells that is really unmatched in biomedical research. But the problem is that our data analysis approaches are often inelegant, poor fits for our data sets. They're like putting lab gloves on our feet, as you can see here. And so I think we've got a more elegant solution for you that we're excited to talk to you about today. One of the problems with classical flow cytometry data analysis is that when you're focused on just identifying a handful of populations in a 20 or 30 parameter panel, you're missing a lot of the information that's built into that data. Moreover, when you're focused only on one or two populations, you end up having to do a lot of gating that's time-consuming and laborious to actually get to those populations, not just in your first sample, but in every sample after that. The other problem, as many of you are well aware of, is that gating is really subjective, right? If you set a gate, if you move that gate ever so slightly, you get strongly different percentages. And from operator to operator, you can have issues as well with gating. And so we have some interesting approaches that deal with that. We're not going to talk about those approaches in depth today, but more importantly, what we're going to talk about is how we can actually access all of the types of cells that are present in your data set. And one way that people approach this right now is that they do machine learning, or they use these visualizations that essentially cluster cells into groups. And so you'll find that there are different islands in these UMAP and t-SNE plots that people commonly present. But the problem is, again, it's really subjective what a real island is, what a real cell population is. And often when you compare these islands, you'll find that there's some subsection of the island, some neighborhood within the island that differs. That's really hard to define, and you don't get a specific phenotype that's actionable and interpretable easily from this type of information. Moreover, you can see that there's no statistics here. You just have to compare plot by plot, and you don't really know how much uncertainty there is in these systems. It's almost like they're a black box. So today we're going to present to you terraFlow, which has some significant advantages over these existing methodologies so you can finally take those lab gloves off your feet. And I'm going to turn it over to Dan Freeman, who is the Chief Executive Officer of terraFlow and the creator of the platform, to tell you more about exactly what we're doing with terraFlow. Dan?
Thanks, Pratip. I'm Dan Freeman. I'm the CEO of terraFlow, and I'm going to start by showing you what the platform actually looks like. So the first thing you need to know about terraFlow is that there's no download necessary, there's no installation, there's no updates. You just go to our website, dash.terraflow.app, and log in with your username and password. What that's going to do is it's going to take you to basically what is a wizard that walks you through the rest of the upload process. So the first thing we're going to do is up here where it says browse, we're going to hit that button and we're going to navigate to our FCS files. So this is from an experiment where we were studying T cell function. So I've gone ahead and pre-exported live singlet CD3 positive T cells. But you could also upload B cells, DC cells, all lymphocytes, or even raw data depending on the scope of your analysis. You're going to see that's going to create this table that has already been populated with the file name, the number of events, and the date it was created. So all we have to do is fill out this last column called class, which is the thing we want to compare in our data. So in this case, I'm going to say, okay, here are patients who responded to treatment. Here are patients who did not respond. Find me the differences between them. Now, we're actually almost done with our upload. The last thing we have to do is click on this channels tab. And now we have a list of detectors in our FCS file and the associated marker names. So all I'm going to do here is uncheck CD3 and live dead because I've already pre-gated on them before I uploaded these FCS files. So I'm going to say this is flow cytometry. I'm going to give it a descriptive title, and I'm going to hit submit. And that's it. That's actually all you need to do to initialize an analysis on terraFlow. And in 24 hours, you'll get an email that says, hello, Dan. Thank you for uploading your files. Click here to see your results. So while our data is processing in the cloud, I'm going to walk through some of the science that makes this type of analysis possible. What terraFlow is doing is it's taking emerging tools in machine learning and combining them with really decades-old fundamental concepts in flow cytometry. So, excuse me. In flow cytometry, we often define cell types by the combination of proteins that they express on their surface. So for example, here's a classic plot where we take CD45RA and CCR7, and using this, we can identify central memory T cells, effector memory T cells, naive T cells, and so forth, because these pluses and minuses mean something to us biologically. So what terraFlow does is take that idea of combinatoric gating and blow it up to a much bigger scale. Because what we do is we take not just the combinations that we're used to looking at, but we take every combination of 1 to 5 markers that you could possibly form within your data set. So if you have 20 protein markers, you can get something like 200,000 combinations. And so what that does is it creates this table where each row is a patient and each column is the percent of cells expressing that phenotype or that combination. And of course, for each patient, we also have a clinical variable like whether or not they responded to treatment. And so now we can start looking for columns whose frequency correlates with clinical outcome. Now, there's one downside of doing this kind of exhaustive approach. And if you already worked with high parameter data, you may know what it is. It's that what ends up happening is that we get thousands of disease-associated cell types, and many of them have the same markers in common, like the ones I've highlighted in green. So if you look at the top 10 cell types here, it's not clear if these are really distinct populations or they're just different ways of describing the same cell type. So what terraFlow does is, rather than pitting these phenotypes against each other, what we do is we create a network that represents every cell type you could possibly look at. So the first level is combinations of 1, combinations of 2, combinations of 3, and so on. Then what we do is we exclude populations that do not exist in our data. So some of these combinations don't actually exist in biological data sets, and so we don't have to worry about them. Then what we do is we measure how well each of these phenotypes correlate to outcome, and we map that information back onto the network. And now we see different neighborhoods light up with high disease associations. So you can see, for example, in the top right, we have CD4 GITR PD1 has a strong association with outcome, as do similar phenotypes that share 1 or 2 markers in common. And then we also see that down to the left, CD8, CD95 has a moderate association with disease status, but it represents a unique set of markers, so it's still biologically interesting. And so what we do is, rather than just picking out the brightest nodes, what we do is we go across the entire network, and for each of these hotspots, we pick the one brightest node to represent that neighborhood. And so at the end of the day, we get a set of explicit phenotypes that together capture all the major differences between our populations. Now, this may look like high tech, but the key thing here is that what we're doing is the same type of analysis that a flow cytometrist would do if they had unlimited time to sit down and gate every cell type that was mathematically possible. The difference is that we do it in 24 hours, and we come back with the key takeaways you need to know about your data. We have a forthcoming cell immunology paper that goes into that algorithm in more detail, but I think our analysis is ready. So I'm just going to go ahead and show you what the actual report looks like from a user's point of view. So remember, we uploaded our data, and in 24 hours, we're going to get an email that says, "Hello, thank you for uploading. Click here to see your results." That link is going to take us to what is basically a 12-page HTML report that walks you through all of the cell types important to your study. And we're going to start with just a simple introduction to the markers in our panel. So the first thing we do here is that for each marker in your FCS file, we're going to provide a short description based on literature. So this is helpful if you're sharing this with, say, with a collaborator who is not familiar with your panel. This is a quick way for them to get up to speed. The next thing we're going to do is we're going to compare the expression of each marker between your patient groups. So here we can see at a quick glance that GITR and HLA-DR and CD272 are elevated in our Hodgkin's lymphoma patients compared to healthy individuals. Now, in Chapter 1, we're looking at individual markers. In Chapter 2, we're going to look at the actual populations driving those patient differences. And this is what the output of that analysis looks like. So you can see that we've identified a list of cell types that differ between our patient groups. And for each of those cell types, we're reporting the average expression in each patient group, as well as a standard deviation. So if it's red, it means it has high expression compared to the other patients. If it's blue, it means low expression compared to the other data sets. Now, what's really interesting about this output is that we're not just labeling these populations POP1, POP2, POP3, but for each of these populations, we're actually going in and defining it with an explicit gating path that you can replicate across batches and across data sets. So we're not asking you to trust our AI implicitly. You can actually go into your standard gating software, gate that phenotype, and see for yourself that it does indeed correlate with outcome. Now, the rest of this chapter is basically presenting the same information in this table, but in different ways. So for example, here we have a classic box plot representation where each box is a different patient group. and each dot is an individual patient. So now we can see really clear differences between our patient populations. And the other thing we do in this chapter is we define a gating tree that combines all of our selected populations. Now, what I really like about this tree is that here, the brighter the node, the stronger the correlation with outcome. And you can see that as you traverse down this tree, as you keep adding markers to each phenotype, the association with disease gets stronger. And that's really the essence of how terraFlow works, because we've already gated every cell type. We know where those disease associations are, and we're just showing you the most direct path that gets you to that disease-associated cell type. Now, I don't have time to hit everything in this report, but I want to show you one more highlight, which is the panel optimization step in Chapter 3. So when Pratip and I were working at the Precision Immunology Center at NYU, one thing we realized is that these larger panels are really useful for exploration. But if your goal is just to predict an outcome, you may not need to stain 30 antibodies every single time. And so what this chapter asks is, what is the smallest panel that I can design that allows me to make accurate predictions of patient outcome? And the way we do that is using a method that in machine learning we call recursive feature elimination. So what we do is we start with a panel consisting of all 24 markers. And on each iteration, we identify and we remove the weakest predictor. So we say, okay, what if we had only stained with the top 23 markers, 22 markers, 21 markers? How low can we go? And what we realize is that as you start removing markers from your panel, our ability to discriminate patient groups remains stable, even improves a little bit until there's a critical set of markers remaining. So in this case, it's the markers highlighted in blue: CD152, CCR7, 69, 127, and CD4. And what we show in our preprint is that we can do a follow-on study where we stain our patients with just these 5 markers, and we obtain better separation than when we use all 24. And so you can imagine, if you are planning a follow-on experiment, what you can do is you can cut those non-informative markers and then backfill with a new set of antibodies. Or if you're planning a follow-on clinical experiment, now you can do these on the lower parameter instruments typically used in clinical settings. Or you can just take this at face value and understand biologically what is really the core features driving my patient differences. So that is the standard analysis that comes out of terraFlow. It's not something I curated from the presentation. That's just what you get in your inbox within 24 hours of uploading data. And now I'm going to hand it over to Pratip to show you a use case that we developed using this technology.
Thanks, Dan. That was really awesome. So, one of the things that Dan and I have focused on in developing this approach is usability, because we found the same problems doing this type of high parameter work for years and years and years. We have difficulties oftentimes communicating the information to people. We have difficulties in visualizing information. We have people with different levels of understanding. Sometimes you have a stakeholder at a company who isn't really an immunologist and might not know all of these cell types. And so we've tried to keep things as simply elegant as possible to do powerful approaches and powerful data analysis using these really interesting technologies in a way that is as accessible as possible. And hopefully you'll see that through this biological example. We have a number of other examples from clients who are spread throughout industry and major academic centers. But this is the one that is going to be in our Cell Reports paper that's hopefully going to be coming out this summer. And so we want to show you a taste of this. So we'll get started with our analysis in action here. And the disease setting that we're looking at is a disease that is a lymphoma, and it's near and dear to my heart because I had it as a grad student. It's Hodgkin lymphoma. It's a cancer of immune cells that affects mostly young people in the most productive years of their life. The historical data that is available about Hodgkin's lymphoma suggests that patients have defective immune systems. So when they're given DTH tests, often they have dysfunctional or lower DTH responses than people who don't have this lymphoma. So we were wondering in this work, what is the cellular nature of this defect? And does the treatment that you get, most of the time chemotherapy, does that treatment remodel the T cell compartment? And do these types of functional defects or changes in the immune system persist after treatment? And so those are all questions that we could answer with terraFlow by comparing patient groups. And so we had 3 different cohorts. The first cohort, actually 2 different cohorts. The first cohort was an exploration cohort where we had 47 Hodgkin's patients and 23 healthy donors. And then, because any good test of a new algorithm really does require an independent validation study, we did that independent validation study with 10 patients to identify the accuracy of the cell types that predict the differences between the patient groups. Among the Hodgkin's patients, we had samples from before treatment, samples from after treatment, and with the healthy donors, of course, they're healthy individuals. We had 2 flow cytometry panels. The first panel looked at just identifying T cells by their CD3, CD4, and CD8 expression. And then we could look at T cell differentiation using CD45RO, CCR7, CD57, and CD95. And then together with those markers, we could look at the immune checkpoints, PD1, CTLA4, OX40, ICOS, and TIM3. Some of these immune checkpoints are ones that inhibit or suppress immune responses. They're markers of exhaustion like PD1 and CTLA4. And others actually amp up T cell responses like OX40 and ICOS. Then we looked at cytokines, interferon gamma, IL-4, IL-13, IL-17, and TNF to provide a functional profile, to provide a kind of functional landscape of the immune response in Hodgkin's patients. The second panel looked at, didn't stimulate the cells. The first panel actually stimulates the cells and stimulates them with PMA ionomycin, a polyclonal activator of T cells, and looks at these functional responses. The second panel doesn't stimulate these cells and looks at them directly ex vivo. Again, we identify the T cells, look at various T cell differentiation markers, look at a few more immune checkpoints, adding now CD244, GITR, and BTLA. And then we look at activation of cells using CD137, HLA-DR. We also look at chemokine receptors like CCR6 and other markers as well, like CD203. And so we use these 2 panels then to investigate these patient samples. And the first thing we did was try analyzing this data using 2 commonly used methods that are pretty advanced and usually require some bioinformatician help, but are commonly used out there. The first is an algorithm called FlowSum. And what FlowSum does is basically identify clusters within your data and then automatically identify those clusters and then compare the frequencies across patient groups. And what you see is that there aren't that many significant clusters here. There are really just a handful of clusters. And when you look at the names of those clusters, they're kind of ambiguous, right? Metacluster 2, metacluster 3, that's not what we typically name T cells, right? So we don't have an exact phenotype. And if we want an exact phenotype, the best we can do is look at a heat map that FlowSum produces that shows the expression of each of these markers. But even so, this heat map is just showing medium fluorescence intensity. And so it's hard to know if something is truly positive or if it's dim, or if it's, it's hard to interpret these heat maps. And you really don't know which of these markers are important, because the heat map shows all of the markers in the panel and the expression across clusters, but you don't know which of those is most important to defining the difference between the patient groups. So ultimately, FlowSum was dissatisfying to us. We tried another algorithm that was reported in Nature Communications. It's called CellCNN. It only found 2 phenotypes that differed significantly between healthy donors and Hodgkin's lymphoma patients. The differences were significant, but again, there are only 2 phenotypes. They're named ambiguously, and you can't identify which markers are most important in driving this difference. So you see that we're running into the same problem. We still have lab gloves on our feet, and it's still kind of messy, and we're slipping around the lab and not figuring out how to do this stuff the way that we really want to look at the data in a complete and comprehensive way. So we put the data through terraFlow, and unlike FlowSum and CellCNN, we're actually able to define strong and explicit phenotypes. You can see that each of these graphics is headed by a real phenotype: CD272 positive, CD4 positive, CD45 RO negative, GITR positive cells. You can actually go to your data and you can gate that, and you can easily replicate this and prove that it's correct. terraFlow is identifying the populations on a massive scale in the same way that you would if you just had a handful of populations to gate. So we have explicit phenotypes, and we have strong associations that show the differences between Hodgkin's lymphoma patients and healthy donors. And one of the things that you see is that the patients who have Hodgkin's lymphoma are in yellow here, and they have higher levels of GITR expressing cells, CD272 expressing cells, PD1 positive cells, all markers of cellular exhaustion. And so we can define some of the immune defects in Hodgkin's patients. We can show that they have elevated levels of exhausted cells in their peripheral blood that include combinations of GITR, CTLA4, PD1, and TIM3 expressing cells. So T-cell exhaustion may underlie some of the immune defects in Hodgkin's disease before they get treated. Before the patients get treated, they have these kinds of exhausted cells that make them different from healthy donors. What's interesting here is that these may be targets for a second generation of immunotherapy. Hodgkin's patients, the standard of care is typically a chemotherapy regimen called ABVD, and that chemotherapy regimen has some significant side effects. People have looked into using anti-PD1 as well, or in different ways in combinations, but there are drugs out there that are in pharmaceutical pipelines for GITR and TIM3 that might represent a second generation of immunotherapy that could help Hodgkin's patients. So we're identifying targets that are interesting to look at therapeutically. But we're still identifying a whole lot of different combinations of populations that are defined by a number of different markers. And this is where our recursive feature elimination platform becomes very important, because it helps us hone in on just those markers that are most important in the difference between healthy patients and Hodgkin's donors. And so when we look at that data, we can see that we build models that include the full set of markers and that have, a certain level of specificity for the outcome. A certain relationship to the outcome defined by the area under the curve here. And then as we drop markers, we can see that the AUC is kind of going up, but then we hit a point where with PD1, after we drop PD1 from the group of markers that we're looking at, suddenly the predictive power, the relationship to outcome really drops. And so you can identify PD1, CD103, CCR7, and GITR as the most important markers to defining the difference between the patient groups. And if you look at the phenotypes that have those, you can identify that in panel B here. You can identify that GITR positive, PD1 positive cells are the ones that are most different between healthy donors and pretreatment and pretreatment Hodgkin's patients. And then when you look at the area under the curve, there's a strong relationship between those populations and the difference between healthy donors and outcomes. So now, instead of having various different phenotypes that we have to parse through that include GITR, CTLA4, PD-1, and a few other markers, now we know that GITR and PD-1 measured together in maybe just a 2-color panel or maybe a 5-color panel that includes CD3, CD4, and CD8, just in a simple panel can tell this difference between healthy donors and Hodgkin's lymphoma patients. And what's important about this is not necessarily the example of comparing Hodgkin's patients to healthy donors. It's your example, comparing non-responders to responders, or comparing people who progress with their disease versus those who don't, comparing a vaccine response to a placebo. Those types of analyses are very, very well suited for terraFlow, and we can identify the cell populations that are most important in those analyses. And in fact, those are the types of analyses that we've done with other groups. Hodgkin's survivors after treatment, 6 months after treatment, when we look at their cells, we still see that they have a persistent exhaustion. They have cells that express GITR, CTLA4, and BTLA, and those are elevated compared to healthy donors. And so you can see that we've really fleshed out some of the natural history of Hodgkin's disease and helped people understand the nature of immune defects and the potential for immune defects in Hodgkin's patients with this example. Again, as I said, this is kind of a toy example to really show the power of terraFlow, but it really, these methodologies really shine when we're looking for differences across treatment groups or patients who respond to therapy or don't respond to therapy. So that's our example of how we use terraFlow, and we're looking forward to showing you with your own data sets and helping you discover everything that you possibly can in your data. I'd like to introduce our team again. Dan Freeman is our Chief Executive Officer. He's got a strong background in biology from our lab at NYU, where we started to develop this. And now he's also been involved in computational science and doing AI work at Harvard. My name is Pratip Chattopadhyay. I'm Chief Scientific Officer. I was at the NIH for many years developing flow cytometry and single-cell technologies. And then I moved to NYU and founded the Precision Immunology Lab, where we identified really the need to have these kinds of data analysis tools, and we started to develop terraFlow. And then Ariel Ginsberg is our Chief Commercial Officer, who has a strong background in clinical flow cytometry and bioinformatics. And her expertise now is bringing exciting new technologies to market. And so you haven't met her today, but over the course of working with us, she's a key member of our team. And so you can see that we've got 3 individuals who are heavily invested and have great backgrounds in doing this type of work and are really dedicated to bringing the best science and the best results to your studies. And so hopefully you've seen here that you don't have to wonder anymore what you're missing in your data. We can take a complete and comprehensive approach to data analysis using terraFlow. And we're here to answer your questions, and you can reach out to us certainly at any time to learn more about the platform and see it in action personally. So I'll leave it at that, and we'll take any of your questions. We'll bring Dan back in for the question and answer session. Again, thank you very much for listening, and thanks for the opportunity to present here.
Well, thank you so much, Pratip and Dan, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. We've already got some great questions coming in here. So our first question asks, how are the population frequencies reported as percentage, as Percentage of all cells or percentage of parent on the tree. The tree implies there is an underlying hierarchy. How is that determined?
Sure, I can take this one. So in that tree you saw, all those populations are reported as a percent of total cells. So in that report, we were studying T cell function. So those cells are reported as percent of all CD3 positive T cells.
And just to add on to that, you can always adjust that number, because if you know the percentage of a downstream population, a subset, you can multiply the two percentages together and you'll get that information as well.
Great, thank you. All right, next question here asks, does this only work for markers that are strongly bimodal, and how is the cutoff for positivity determined?
Yeah, I can take this one because I see a question that's perfect for Pratip next. But basically, there's two ways you can define positive and negative in terraFlow. The first way is using the classical threshold approach. So in that case, you can set your own threshold using an FMO, for example, or you can leave it blank and let terraFlow choose a threshold for you based on that bimodal distribution. The other way terraFlow analyzes data is by treating marker expression as a continuum. So in that case, we are reporting markers as bright or dim as opposed to, and terraFlow actually does the entire analysis both ways, and then it combines it into one report. So we haven't covered that here, but I'd be happy to meet with you in a follow-on call and show you what that continuous non-gating approach looks like.
Yeah, and it's totally, sorry, Kaylee, not to jump on you, but the non-gating approach is a really exciting approach that we did not talk about today. And for those of you who might be interested in that, it's not automated gating, it's different, and we'd love to walk you through that. So I think Alice had asked this question, and we'd love to get on the phone with you and talk a little bit more about it.
Great, thank you. All right, next question here. Can it handle discriminator that is non-binary, greater than two groups, or continuous, for example, disease activity score instead of responder versus non-responder?
Dan, do you want to take that?
Yeah, I'll take this one. So it's right now we only do we compare patient groups. So that could be pre versus post, healthy versus disease, responder versus non-responder. We do support comparisons of 3 or more groups. We support paired data. So for example, pre and post samples from the same individuals. And we also support batch data. So for example, if your data was collected in multiple plates, you can actually input the plate number in that table, and we factor that into the statistical analysis.
Great, thank you.
And so, the other thing to think about here is, oftentimes when you have a continuous variable that you want to compare to keep things simple, you still end up moving to, a group by group comparison. And so, oftentimes you'll see that there's a lot of value in the group by group comparison for simplicity's sake. We have on our, hopefully to-do list, or something that I've been hoping that we would do, is survival analyses and things like that. And so those are continuous variables that I think will be folded in future versions of terraFlow.
Yeah, and I'll just add one more point, is that the reason we don't have continuous variables or survival analysis is that we just haven't come across a data set that requires that feature. So if you have a really compelling use case for a continuous variable or survival plot, we want to have that conversation with you, and it very well may become part of the platform based on that.
Great. All right, now what is the biggest benefit that your users have seen with terraFlow versus other types of algorithmic approaches?
So that's a great question. I think the biggest benefit is time savings. It's just literally a matter of uploading your data to the cloud, having it, and then it's processed, and you get this incredible report that has presentation-ready graphics and is simple enough. It's described in terminology that is understandable to anybody with a scientific background. So they don't have to be an immunologist. They don't have to be a bioinformatician. They don't have to have any specialized knowledge. And because of that, you can easily share this report with other stakeholders. That could be your clinical collaborator. That could be your, a senior VP in a pharma company. And because of that, because you don't have to reinterpret the information, there's a huge time savings. Compared to other algorithms, terraFlow is inherently understandable. It's based in the language that we have always used in flow cytometry. You saw that in the talk a little bit. It's based in populations that are positive and negative. It reports directly precise phenotypes rather than islands or clusters or sets of dots or things on a heat map that require a lot of vague description. And so I think those are two of the biggest benefits that we have for the platform. And the reason that we developed the platform that way is because that's exactly what we were frustrated with. Every time we had a data set, we had to get in line with the bioinformaticians, and it was great to work with them, but we had to get in that line. We had to make sure they understood flow data, and then we had to take the information that they gave us and reprocess that into something that could be communicated and easily interpreted. And so we're giving you all of that in one package, in one analysis that comes to your email inbox within 24 hours, which I think Dan and I are really proud of that.
Great, thank you, Pratip. All right, we've got time for a few more questions here, so we'll continue. This next one asks, can you only compare two conditions? What if there are more than two?
Yeah, so you can definitely compare three or more patient groups. So for example, we just did a study where it's a pre-treatment, post-treatment, and also healthy controls, and you see all three patient groups side by side.
Great, thank you. Now, is the platform workable with spectral files, and can it parse the metadata?
So it is workable with spectral files, and my other endeavor is a contract research organization called Talon Biomarkers, and we have been generating data. We've been generating data that is spectral flow cytometry data and using terraFlow to analyze it. So it's totally workable with spectral. It works really, really well, and I've been very happy with it. In terms of metadata, I guess what you're asking there is if it can automatically pick out a keyword from an FCS file and use that to populate the groups. We don't have that feature built in yet, but we can do that. I think one of the things that Dan and I are very proud of also is that we are responsive. We're looking to make the software as usable as possible.
Great, thank you. All right, now how do you deal with batch effects? Can you compare data from different instruments, and do you normalize the data?
Yeah, thank you so much for asking that. So I mentioned this briefly, but we do handle batch effects, and that is one of the variables one of the metadata variables you input in the analysis. So if you remember when I was uploading the data, there was that one column where I was typing in responder versus non-responder. Basically, what you do is you push a button, and another column pops up where you can input the batch that sample belongs to. For example, plate or date of experiment. And the way we handle those batch effects is we don't normalize your data. We don't actually manipulate your FCS files. Rather, what we do is we run the entire analysis independently within each batch. And then we combine them and look for disease associations that are consistent across the entire data set. So we're not manipulating your data, we're doing a statistical analysis to look for robust differences.
Great, thank you, Dan.
And we could also add here that our non-gating approach, not that we want to bring that up too much, might actually help with batch effects. And so if you have a project, a data set that has lots of batch effects, we'd love to play with it and see if it's a problem. All the flow cytometry I do is perfect, so we've not had that access. I'm of course kidding, but yeah.
Yeah, and I just had one more point about this question before we move on, is that, I mean, the whole point of our analysis is that we bring this back into a cytometrist's own language so that you don't have to trust the AI implicitly. So for example, when we say T cells that are predictive of GITR PD1 GITR PD1 T cells are predictive of a drug response, you can go into a typical gating software, you can gate GITR PD1, and you can replicate that using data from any instrument, from any data set, from any fluorophore. And so the whole point is that we're giving you these simple phenotypes that you could replicate on your own without having to get your data exactly the way a particular machine learning model wants it to look like.
Great, thank you. Now, can you tell the system factors that are not included in the FCS file, such as patient glucose levels upon sample collection?
So you can always do that. You can have that be another variable. One of the things that we've done with our data sets at Talon is that we've run one comparison through terraFlow, and then we've gone back and set up an additional comparison and run that through terraFlow. And so that's how we would approach this. We would probably use it as a second separate analysis. So you can imagine that, let's say you had patient glucose levels, and then you also had disease progression. Well, you look at disease progression first, and then you identify the populations that are associated with disease progression, and then maybe you look at glucose levels separately and see if they're the same populations and begin to interpret it that way.
Great, thank you. All right, next question. Could you post links for any references describing your methods?
Yes, so we have a,
go ahead, please.
Okay, we have a bioRxiv paper that's out there, and we have a paper that is in revision at Cell Reports that is basically the bioRxiv paper updated somewhat with reviewer comments. And so hopefully that'll be through the review process in the next 2 to 4 weeks. And then we can update the bioRxiv paper to reflect what's in the draft that's in Cell Reports. So stay tuned, reach out to us for any references. At Cyto, at Cyto 2024, we'll have a poster on an analysis that we did with a public COVID data set. And so whoever asked that question, if you're at the Cyto meeting, look for us there. And we'll be happy to go over that data also with you. I have some data from Talon's work also that I'll have to ask the client if we can share that. But we've used it a fair bit, and we have academic customers as well who have been using it. We'll ask them for permission to share their experiences with you, but we're happy to have you see it in action.
Great, thank you, Pratip. Now we've got a question in here asking about cybersecurity. Do you have cybersecurity?
Yes, great question. So we definitely built this platform with cybersecurity top of mind. So it's a cloud-based software. The entire analysis lives in AWS, which is a lot of enterprise companies already have experience working with. We've also completed a SOC 2 Type 2 cybersecurity audit, which is one of the most comprehensive audits you can do in the industry. Basically, an outside person comes in and verifies that you adhere to all the industry standards. And our experience, that makes the onboarding process much easier for our clients. So we've been down that road with pharma and with academic users, and we know how to make the experience as painless as possible.
Thank you, Dan. All right, it looks like we have time for one more question here, so we'll wrap up after this one. Do you have minimum recommended marker counts to find your algorithm useful?
I can take this one, and we'll go to you, Pratip. There's no minimum here. I mean, really, this is a platform where you have data and you want to find some differences between your patient group, and you don't want to do it by sitting down and gating every cell type that is mathematically possible. And so most of our clients have in the range of, I'd say, 12 to 20 markers, sometimes a little bit higher. But we've also done data sets with as few as 8 markers and have found useful insights that might have been missed using existing methods. And so there's no minimum to the number of markers you can upload to the platform.
Great. Well, that will conclude our session for today. So I want to thank you again, Pratip and Dan. Do you have any final comments for our audience?
So I'll let Dan conclude, but just before we conclude, first of all, thank you, Kaylee, for moderating this. This has been really great. And we're so happy to get out there with this platform and to talk to you all. There are some questions that are outstanding that, talk about price and reference values and things like that. I would encourage you guys to reach out to Dan, me, or Ariel Ginsberg, who is our Chief Commercial Officer, because the three of us are happy to talk to you about those, and we'll probably reach out to you by email to give you those answers as well. One of the things, again, that I wanted to highlight is that this was really an effort that came out of our own needs doing immune monitoring and immune analysis. And so I think that's the incredible value here, that we're people who understand informatics, understand immunology, and understand flow cytometry. So thanks again. Dan, do you have any comments? I'm going to wrap us up here.
Yeah, I'll just follow up this point is that, I think one thing that makes our company unique is that, Pratip and I and Ariel, the three of us are all flow cytometry people. Like, Pratip doesn't like to brag, but he actually ran the first ever 30-color flow cytometry experiment. I grew up in science, so to speak, with a desk next to a fax symphony instrument. And so we basically built a platform to analyze the data that we analyze data, but much faster and in a much more comprehensive way, in a way that felt intuitive to us. And so it is such a privilege to be able to talk to you guys about this technology. And I know there are many questions we weren't able to answer, so please follow up with us. We'd love to continue the conversation with you one-on-one. Oh, and if you are going to Cyto this year, we're going to have a workshop, a poster, and a technology workshop pitch at the opening plenary. So I hope to see you there.
Yes, very exciting. So thank you again, Pratip and Dan, for your time today and your important research. We would also like to thank Labroots and our sponsor, terraFlow Bio, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.